So let me start off by thanking you for giving me the opportunity today to give a very brief, very fast overview about what's going on in our laboratory with some published and unpublished work. We're going through an immense amount of material very quickly. So if you have any questions, if there's anything you'd like to follow up on and discuss on any of these research projects, please feel free to email me at nlac@prostatecenter.com. So my laboratory is almost exclusively focused on understanding prostate cancer. And we're interested in this disease because it's a remarkably common disorder, with 1 out of every 8 Canadian men being diagnosed with prostate cancer in their lifetime. Now, to better manage and treat this disease, our group is broken up into 2 parts. One of them is a pure basic work where we will take advantage of all the new experimental techniques that are available to us, whether it be high throughput assays and multi-parallel reporter assays or single cell approaches or CRISPR screens. And then using these kind of really high throughput cutting edge approaches, we can then kind of tease apart fundamental basic signaling pathways in prostate cancer to really get down to how this disease kind of initiates growth, proliferation, and then resistance. The second arm of our laboratory is far more translational, where we take all this information that we've gained, all of our understanding about the mechanism and the basic biology, to identify novel therapeutic targets and then eventually develop small molecule inhibitors that target these using kind of classical in silico pharmacology. Now, a big part of our basic work is focused on a signaling pathway called the androgen receptor mediated transcription. And this is by far the most important signaling pathway in prostate cancer. The vast majority of both primary and even late-stage resistant prostate cancer is totally dependent on AR signaling. And given this kind of critical function and really important role, there's been quite a bit of work to understand how the AR works. The first part of the activation pathway we've got down pretty cold. We know this inside out about how this works. Where the androgen receptor will be circulating in the bloodstream, and then eventually it'll bump into a prostate cell where it'll passively diffuse into the cell, and then bind to an androgen receptor protein that's held in an apoconformation in the cytoplasm. Following binding, there's an allosteric modification that causes the androgen receptor then translocate from the cytoplasm into the nucleus, where it then binds onto androgen receptor binding sites on the DNA. And it binds to a lot of these. There are tens of thousands of androgen receptor binding sites in DNA. Once it's on the chromatin, it then induces transcription. But my argument would be that while we understand that first signaling pathway really well, while we understand the activation really well, we don't understand the effector mechanism at all. So once the androgen receptor is bound onto chromatin, we can identify these sites very easily using kind of current functional genomic methodologies, but we don't know how they contribute to gene transcription, largely because we don't know what they do on DNA. And I would argue that only by really understanding something truly can we effectively target it. So we have spent a lot of time in our group teasing apart exactly what these different elements are doing. What happens to the androgen receptor once it's on DNA, and how does that in turn then impact transcription? In a recent publication, we functionally mapped all the kind of classical androgen receptor binding sites identified from clinical prostate cancer to give a really detailed locus-specific resolution information about how exactly or which of these sites exactly are AR-driven enhancers. And this information was conducted all in vitro, but the results we generated correlated very well to clinical in vivo samples. And having this map actually proved to be very effective to then begin to identify which sites were likely very critical for gene transcription and potentially very critical for prostate cancer progression. In a previous publication, we had shown that the sites where the androgen receptor binds, the actual sites on the chromatin where the AR binds, have a large enrichment in mutations. There's a big spike in somatic variants, particularly single nucleotide variants on chromatin. Now, what we didn't know, though, was were these non-coding mutations simply correlative because the AR was binding there, or could they cause increased growth and proliferation? Could they act as driver mutations in there? So given the large number of non-coding mutations, given the large number of sites, this was not really a technically feasible step at the time. So what we could do then is take that functional map we generated and then overlay these non-coding mutations to identify some for functional testing. And when we did this, we observed very clearly that some of these mutations had a profound impact on androgen receptor enhancer activity. and that they would impact the gene transcription of tumor suppressors and oncogenes, potentially giving functionality to some of these non-coding mutations. And what we learned from this experience was something quite surprising, something that we didn't quite expect, where we can take these methodologies and then we can start to apply it to really all non-coding mutations. Simply because that non-coding mutations, particularly enhancer mutations, are very amenable towards high throughput screening. The functionality of enhancers are very kind of similar and common, so that you can take a look at multiple enhancers in one assay and look at it. This is not the case in a protein coding mutation, where each protein coding mutation, you can identify them very easily and you can kind of predict what the impact may be. But at the end of the day, you have to have a specific assay for that specific protein. And that if you move one protein over, if you test a different protein, you have to develop then another assay to look for the protein activity. And this kind of really limits the throughput available to you to understand how somatic mutations in protein coding space impacts cancer development. But because enhancers have a common functionality, you can then test all enhancer mutations in a single go. So we spent a lot of time recently developing experimental methodologies to functionally test non-coding mutations and determine their impact on cancer initiation or enhancer functionality or gene transcription. So we've got some recent publications that demonstrate the utility of this to then look at kind of single nucleotide polymorphisms that are associated with prostate cancer progression, and also some of the somatic variants that kind of turn into potential driver mutations. While this works very well, one of the limitations is that we needed to essentially find a sample that has the mutation already, and that kind of is not terribly high throughput. So we've developed the methodologies and to do complex targeted mutagenesis, where we can take pools of these MPRA assays and then introduce in the targeted mutations we want to functionally characterize non-coding somatic mutations. Now, this could be also applied to protein coding space as well, to look at kind of combination of mutations or different proteins all in one experiment. In addition to kind of increase the throughput of some of this assay, we've modified our approach to be amenable to single cell enhancer method development. So we now have approaches that can then quantify the exact kind of changes at single cell resolution in the enhancer activity at these androgen receptor binding sites. And this is really a promising methodology to tease apart the functionality and the diversity of the enhancer usages by the androgen receptor. But as you can tell from all these approaches, this is extremely basic. This is an extremely kind of fundamental understanding about how the androgen receptor works. This, in addition to kind of some large-scale CRISPR screens, have then guided some of the second aspect of our work in the more therapeutic angles, where we've been looking to identify novel therapeutic targets to treat prostate cancer. And we do this simply because there's not a huge amount of diversity. There's an increasing amount right now. We've got some new tools that we didn't have before, but as a whole, the vast majority of therapeutics that are used to treat late-stage prostate cancer are all built by targeting the androgen receptor. So I have a list right here of all these different kind of therapeutics, from enzalutamide to daraglix, to bicalutamide to darolutamide. All of these kind of AR pathway inhibitors or AR signaling inhibitors, they work by targeting different parts of it, but they all have that same common AR target. And as an academic group, I thought that we should have the ability to go and look for novel targets. We should be looking for new things in addition to androgen receptor to potentially treat late-stage prostate cancer. Now, when we're looking at these targets, we're looking for really two things. One of them is that an ideal therapeutic target is extremely important for the progression of prostate cancer. So this has to be something that is essential in prostate cancer, that is absolutely required for the growth and the proliferation of the disease. But it also can't be broadly nonspecific. We're not looking for the bleach phenomenon. We want to find a therapeutic target that is essential in prostate cancer cells, but not essential in other kind of normal cell types. So you would have limited side effects associated with it. And taking advantage of large-scale CRISPR screens and some large-scale methodologies, we dug through the data. We kind of found all the information we could to identify potential therapeutic targets that would give you selectivity and essentiality. And from this, one that we've become very interested in is a protein called HOXP13. It's a member of the homeobox protein that is typically associated with development. But unlike the vast majority of these HOX family members, HOXP13 remains highly expressed in the adult human prostate tissue. And it's largely found almost exclusively within the human prostate. It's not really found in any other site except for the large intestine. But what's interesting is that when we test these things, when we look at the impact of HOXP13 on PCA growth in vitro, it is remarkably essential, regardless of the androgen receptor status. So AR positive, AR negative are both affected by HOXP13 loss. And this is particularly powerful because then it suggests that HOXP13 is not an AR-driven effect, that what you're seeing here is not via AR, it is independent of AR, giving you a totally new avenue to potentially target late-stage prostate cancer. We can also see the same phenotypes in vivo, where if we take a guide RNA targeting HOXP13 and we look at it in these in vivo models, whether they be AR dependent or independent, we see that loss of HOXP13 has a profound impact on growth and proliferation, but has almost no effect on the normal tissue or normal development. So we have this kind of remarkable essential role within prostate cancer development. And then when we go and look at the DepMap data, where there's 1,000 cells that have been tested, we find that only primary or prostate cancers are affected by HOXP13 loss. So you have selectivity as well. You've got those two parts you're looking for. And we showed very clearly that the HOXP13 activity is not AR associated, where in an AR negative cell, there's almost no different or no overlap between the binding sites, suggesting an independent mechanism. So we've got a lot of ongoing work around HOXP13, teasing apart the mechanism of action of all of this, but also to develop novel inhibitors. So as a broad overview, these are some of the topics that my laboratory is working on. We have other projects that we're particularly enthusiastic about. And if anybody who's watching this video has any interest, please feel free to reach out to me. Thank you very much.